Thank you. Good afternoon, ladies and gentlemen. It gives me great pleasure to welcome you all to this auditorium today, and to those of you who are not joining us in the auditorium but are joining the web stream. We can't see you. I hope that you can see us and will be able to participate in what's going on. Let me introduce myself. I'm Tony Hardy. I'm an external expert from the UK. I have worked in UK science for the government on the impact of agriculture on the environment and pesticides and food safety for my working career. I've worked with EFSA since it was formed in 2002. I've chaired the pesticide panel for 9 years, and I'm currently chairman of the scientific committee. I would also like to welcome my co-chair, Professor Jean-Louis Bresson, and ask him if he would introduce himself, please.
Thank you, Tony. Well, I'm a pediatrician working at Necker Enfants Malades, otherwise Sick Children Hospital in Paris, and I'm professor of nutrition at Descartes Medical School. As you can guess, I'm pretty much interested in infant and child nutrition, but as well into the nutrition during pregnancy and lactation. I'm also an external expert working on the NDA panel, which means nutrition.
Thank you. In this exciting session, and I hope it will be an exciting session, we've got a very interesting and challenging program for you. I'm joined on the stage with 4 distinguished speakers. Dr. Anne Salonen from the University of Helsinki, Professor Per-Luigi Nicotera from the German Center of Neurodegenerative Diseases, Professor Richard Sharp from the University of Edinburgh in the UK, and Dr. James Trusco from Michigan State University in the States. Shaping the Future of Food Safety Together is the overall title of this conference, and this particular session is to continue to think about some of the themes on science, innovation, and the society, and again to look forward as to where the science is developing and what are likely to be its implications on food safety. The 4 topics that cover areas which are the subject of major scientific research and development and are expected to impact the development of regulatory assessment methodology. And the themes we're going to be talking about today are intestinal microbiota, neurodegenerative diseases, reproductive endocrinology, and epigenetics. The format of this afternoon will be that each of the 4 speakers will have 30 minutes to make their presentation, and then we will have a 10-minute technical questions for clarity. At the end of the session, we will try and draw it together for 20 minutes into a panel discussion. And at the end of that, we may or may not, if it's very difficult, try to add a few concluding comments on how the session has gone. There's only one house rule, and I'm sure you've all obeyed it, but if you have a mobile phone, please could you either silence it or turn it off. Thank you. It therefore gives me very great pleasure to introduce our first speaker. Dr. Anne Salonen is a senior researcher and docent at the University of Finland. She has multidisciplinary training in biosciences and a PhD in microbiology. Her research activities are focused on the composition and activity of the intestinal microbiota in health and disease, with special interest in reciprocal interactions between diet and the gut microbiota. Dr. Salonen will speak about food and health, the role of intestinal microorganisms in human health. Please, Dr. Salonen, the floor is yours.
Good afternoon, ladies and gentlemen. I would like to thank EFSA for the opportunity to represent gut microbiota field in this conference. Gut microbes have moved center stage in biomedical and nutritional research during recent years because of their central importance for our health. And they also represent one mechanistic link between nutrition and health. When I talk about microbiota, it's basically a new term for normal flora, and I use interchangeably also the term microbiome, which has, in addition to referring to the microbes themselves, has also a functional aspect because it refers normally to the genes or functions these microbes carry. So these microbes are an integral part of our body. Actually, numerically speaking, they outnumber our own cells because for every human cell, we have 10 resident microbes in our body. And in the gut, they represent hundreds, if not thousands, species. Altogether, these microbes encode something between 5 and 10 million genes, meaning that the genetic capacity of our gut microbiome actually outnumbers our own genome by a factor of 100. And we have really evolved to live with these symbiotic microbes so that some very fundamental functions of our body, such as digestion or immune functions, depend on these microbes. And comprehension of this symbiotic view of life has really changed the attitude towards bacteria in medicine, because previously it was very pathogen-centric and it was thought that the only good microbe is actually a dead microbe, and there has been really a paradigm shift during the recent years, because now also medical doctors acknowledge that actually also a lack of bacteria can cause a disease. Aside of this numerical predominance of the microbial genes in our body, the microbiome also has some other features, really intriguing features, that explain why it's a center of such an active research. And unlike our own genome, which is first of all highly similar between all of us, and we born and die with the same genome, in the context of the microbiome, actually it varies heavily between the individuals, and even in adulthood we can change this microbiome, for example, via diet. So it's plastic, and we can even transfer it from individual to another in treatment purposes, and this is a highly efficient treatment form, the fecal transplantation, to treat the antibiotic-associated Clostridium difficile infection. So basically, it means that we humans are composite organisms, so that in biological terms, not only our own genetic makeup, but also the makeup of our bacteria make us who we are in terms of, for example, our capacity to digest food, and that of course affects our disease risk. So why have we evolved to carry more than a kilogram of bacteria in our gut? These bacteria assist in digestion. They provide us functions that our human enzymes cannot do. For example, the digestion of dietary fiber. And in addition to releasing energy from otherwise useless dietary compounds, by utilizing, for example, dietary fiber, they release a lot of bioactive compounds that have wide and profound systemic effects in our body. And I will elaborate this soon. But to understand actually the full dimensions of the gut bacterial effect in our body, we have to recall that our gut is not just a digestive tube, but actually it's the main immunological organ in our body, and also it's surrounded by hundreds of millions of neurons. So regarding immunology, the central question in microbiome research is that why we modern individuals who are relatively free of all infectious diseases are actually so prone to get allergies, inflammatory and anti-inflammatory, other immunity diseases. And there is a strong accumulating evidence that actually the bacteria, or sometimes actually the lack of gut bacteria, contribute to these diseases, because especially early in life, when the immunity is developing and our body must learn to kind of find a balance between tolerance and reactivity, it's been shown that the bacteria in our gut have really crucial role on educating the immune system to find a balance. And of course, for example, just to mention that because it's very critical that we can separate this vast mass of bacteria from this highly active immunological tissue, these bacteria also help us to fortify the intestinal barrier. So we agree that intestinal bacteria have a very wide effect on our systemic health. And then it's not surprising that nowadays there are maybe almost 30 diseases where alterations of gut microbiota have been associated. And these diseases, they really range from metabolic maladies to inflammatory diseases, allergies, even neuronal diseases. And I want to make a point here that in the case of humans, the data is mainly associative, because for experimental limitations, it's actually quite difficult to address causality. But from animal models, we know that in many cases, such as in obesity-associated insulin resistance, for example, the microbes really play a causal role, and I will show you later some data on that. A feature that characterizes these diseases where microbiota alterations have been detected is that actually presence or absence of a single bacterium has a little effect. Rather, we typically see a kind of eco-catastrophe, even if you like, a kind of a functional aberration, often also called dysbiosis, which means that the altered ratio of our normal bacteria cause a disease. And that, of course, makes a strong conceptual challenge, because if we talk about microbe-associated disease, which doesn't involve a pathogen, it really has some challenges to show, for example, the causal chain. But actually, this is analogous situation to dental caries, where it was shown already 10 years ago that actually it is not just Streptococcus mutans that can cause caries, but actually any bacterium that can bring the critical functional trait can cause the disease. And we are talking about similar phenomena in the gut. So it's evident that gut bacteria represent basically an independent novel disease risk, and that's why it also has, of course, some obvious therapeutic implications. So if we acknowledge the fact that these bacteria can either act for us or against us, how can we shift this balance? This is a non-conclusive list of factors that are known to affect gut microbiota. And as you see, many of them are actually inherent to our modern lifestyle, that even if we know that, for example, very high hygiene or use of antibiotics is not good for the microbiota, we cannot do that much. However, diet is something we know that we can make individual choices. And I will use the rest of my talk to discuss the interactions between diet and gut microbiome and how that affects health. But before going there, I just want to very briefly to show you also some basic things, because what is modern microbiology? Because the fact is that we cannot culture most of the microbes that are in our gut. And previously, it was thought that E. coli and lactobacilli, which are very easy to grow, that they are the dominant organisms. And now we know that they represent basically just 1% of the microbiota, because we have revealed by using molecular methods, mainly based on DNA sequencing, this majority, which was previously completely unknown. How do we study this unknown majority? It depends on a question. Basically, there is an array of so-called omics technologies, meaning that we target the entire microbial community at once. And normally, if we talk about healthy individuals, it's often a fecal sample, because collecting mucosal microbiota requires an invasive biopsy. And the most common way is to do a so-called taxonomic analysis that allows us to ask that which bacteria and in which ratios they occur in the sample. However, the field is moving towards functional analysis, which allow us to ask that what actually are these bacteria doing? And this is highly, highly relevant, because we think that for the pathologies, the functions are definitely more important than the taxonomic composition, but there are quite some technical challenges for these methods, so that it's kind of early days. And I would say that still over 90% of the studies that are being published are based on this taxonomic analysis. And of course, in some cases, when we want to sequence the entire gene, get the gene catalog of all the microbial genes in the gut, then we can do shotgun sequencing. What is common to all these methods is that they produce massive amounts of data, and the field has become really computationally intensive, and all the time new bioinformatic algorithms are developed, and there are also quite some challenges still also on the bioinformatics side. I'm not going to bore you with bacterial names, but I just want to show you an overview of a typical European adult, of the bacterial content on a very high level, on the phylum level. And also I just want to make a point that unfortunately, for the majority of these bacteria, we actually cannot categorize them yet for being beneficial or harmful. And you see that there is quite some variation, meaning that between individuals, all these proportional shares have been detected. And in fact, each of us has a unique microbiota. And this is not just limited to the composition of the microbiome, it's even the functions, the proteome, as we showed recently. This high individuality possesses some practical challenges, because you can imagine that if we are all different, how do we define the healthy microbiota? And this is in fact one of the largest challenges in this field, because there is a great compositional variation and we still don't understand what all factors affect on that. However, there is much less variation on the functions, and that's another reason why we are trying to actively develop, to put the research more to analyze the bacterial functions. However, there are different ways how we can reduce the dimensionality of this of the variation. And for example, a few years ago, there were the enterotypes were introduced, so that they are different groupings of bacteria based on the dominant organisms. And we ourselves have tried to understand the complexity, so that instead of focusing what is different between individuals, we focus on those microbes that are shared between all of us, the so-called core microbiota. We also use ecological terms to describe the microbiota, because in the end, it is an ecosystem and a complex one. And one very useful term is diversity, which basically means that how many bacteria and how evenly they are distributed in the community. And as you see, there is a strong, the diversity depends heavily on age, so that basically when the baby is born, he or she gets majority of the microbes already during the birth from the mother, and then later on via the breast milk, and of course from all caring persons. And what is noteworthy is that this decline of diversity it's one of the very most conclusive findings in relation to disease, so that people who are not healthy, they have a decreased microbiota diversity. And I just want to make a point before I go to diet that that when we talk about the developing microbiota in the early life, antibiotics are actually maybe the greatest threat, that even though they are sometimes lifesavers, the high use in our modern society possess a clear threat for the development of the microbiota. We, for example, have shown that the recent use of, especially of macrolide antibiotics, severely interferes both the diversity development of kids as well as the maturation of microbiota, which in comparison what normally happens on that age. So then we move to the diet, and a very quick mechanistic view. Basically, diet can either modify the gut microbiota by modifying the existing community or bringing new microbes. And I'm not going to detail for the sake of time. We have discussed this in a recent review, but I will elaborate this modifying the existing community by providing substrates. That means that providing food for the microbes. And there is also there is very active research on prebiotics, which is basically the food for bacteria, as well as on probiotic research. And I have to mention that so far EFSA has actually rejected all the health claims, and mainly for the reason that the endpoints have been too generic, boost immunity or improve gut health, so that it's difficult to measure. And also the findings it has been difficult to generalize them to healthy population, because they are typically done, for example, in a case of diarrhea. So when we take a look on what's available for our gut microbes, basically they will eat what we eat, and then the leftovers that are not taken upper in the GI tract, because majority of this bacterial mass resides in the colon. And normally the majority of food that gets in the colon is dietary fiber, because we don't have enzymes to degrade that. Bacteria, in turn, they cooperate on different trophic levels, so that there are a variety of food webs, so that they digest the polysaccharides, and the major output, what comes from bacterial fermentation, are short-chain fatty acids. And I will elaborate these just in a second, because they are very important for our health. If protein is eaten in excess, so that part of it reaches also to colon, gut bacteria do ferment bacteria, also protein, and depending on the amino acid composition, they produce a variety of products which are actually harmful for us. Basically, branched-chain fatty acids, different nitrous and sulfuric compounds that have been implicated, for example, in colon cancer. Not much is known about the interactions between gut microbiota and fat, but it's well known that the bioavailability of many polyphenols actually depend on bacterial transformation. So basically, it means that when bacteria ferment the food we eat, they produce different types of metabolites that reach our body systemically, and in the long run, our bacteria will adapt to that diet it's chronically exposed. And that's one evidence for this comes from the comparative studies between rural and Western societies. It has been repeatedly shown that, for example, comparing kids between Africa and Italy, comparing adults between Africa and Italy, and comparing also Americans to microbiomes of people living in Africa or southern America, shows that there are distinct differences, and the major finding is that we Western people have a reduced diversity in our gut microbiota. And the fact, of course, is that in the rural areas, people eat a lot more dietary fiber. And that has led to a hypothesis that this reduced gut microbiota diversity in Western societies actually reflects a kind of loss of old friends, with an idea that these rural microbiomes represent kind of ancestral microbiomes. And this is something that could be then one explanation why we have so many lifestyle-related diseases, such as the obesity-related metabolic aberrations. And actually, these short-chain fatty acids, they are really multipotent molecules, because in addition to providing energy from dietary fibers, they produce a lot of molecules that either promote our intestinal homeostasis and overall immunity, and most importantly, they are also very strong anti-inflammatory and anti-carcinogenic compounds. So it's easy to understand that if we are very short on this, it can have a profound effect in our gut, in our health. And actually, exactly this is what we think that happens when, with the consumption of energy-dense, fiber-scarce Western diets, that the shortage of fibers, which decreases bacterial fermentation, is maybe one mechanism why we kind of, first of all, that why fiber is protective, because if the typical Western diet contains a lot of sugars that are already easily digested in the small intestine, and kind of put it to simplified way, basically the bacteria in our colon starve because they don't have enough these complex carbohydrates that would come from the diet. A completely independent mechanism that relates the Western diet to gut microbiota-mediated diseases, especially on obesity-related diseases, relates to the fact that the landmark work by Patrice Cani and others later have shown that the high-fat diet dramatically increased the permeability of our gut. And this leads to leakage of bacterial components to systemic sites. And it has been very convincingly shown in my studies that this is definitely one trigger of chronic low-level inflammation, which typifies obesity and leads to development of insulin resistance. There are also other examples how typical features of Western diet or to put that way, that where gut microbes actually mediate the negative effects of Western diet. And actually there are even further from the macronutrients, these food additives, which are quite common nowadays, also pose a threat to our gut microbes. Because it has been shown that, for example, the commonly used dietary emulsifiers strip the protective lining in the gut and can, by that means, contribute to inflammatory conditions and adiposity. And similarly, kind of counterintuitively, the non-caloric artificial sweeteners were recently shown to contribute to glucose intolerance in mice. And the causal contribution of gut microbes was shown in this study, so that, and that's been actually quite a common approach, that gut bacteria are taken from an individual who has a disease phenotype and transferred to naive mice. And if the phenotype can be transferred by transferring bacteria, it must mean that the bacteria are causally related to this disease. So just giving one example of intervention studies in humans, because this basically reproduces the epidemiological studies and also the studies in mice, showing that in this study where healthy individuals were either exposed or consuming either completely animal-based diet or the diet based completely on plant products, so that in the animal-based diet there was, of course, it was basically almost entirely fat and protein, that already in 2 days the gut microbiome changed quite dramatically. First of all, there was much larger change in bacterial functions, again, compared to the composition. And maybe somewhat surprisingly, in this case, it was not the fiber, but it seemed to be really the protein and fat that made the largest change compared to the baseline. And actually, this study reproduced the hypothesis that the high intake of meat and fat, first of all, it reduces the production of these protective short-chain fatty acids, and it was what I had no time to talk about today. It also basically made threefold higher content in the colon for secondary bile acids, which are very strongly implicated in the development of colorectal cancer, as well as these sulfuric compounds, which are also implicated in inflammatory and carcinogenic processes. So we have a $1 million question, then what to eat to make your microbiome healthy and to nurture it optimally. There is both an easy answer and a difficult answer. The easy answer is that, as I have been telling you, that basically what is healthy for yourself based on these studies is also healthy for your gut microbiota. But I want to stress the fact that we don't have sufficiently detailed info to design such kind of dietary compounds that would allow a targeted manipulation for certain health benefit. Because we have some fundamental questions still that we cannot answer. First of all, there is even, as I mentioned in the beginning, for majority of the bacteria, we don't know really are they healthy. So it's a bit unclear what all functions of bacteria we should target. And a major issue is also that because these bacteria digest food in a consortia, that how can we selectively stimulate a beneficial bacteria, because also other bacteria will be affected. And a major point is that what is, I think, really important for the regulatory authorities as well as companies making functional foods, that the microbiota change as such is not a health effect. It can be a secondary outcome, but of course the clinical effect coming from the microbial effect will be the primary readout. And there, as I mentioned, that currently mainly the data is mainly from animal models and from in vitro studies. So there really needs to be more research on humans to address the causality. And the final thing I want to very quickly address is that we all know that even if we eat the same food, we don't have the same health outcome, either the benefit or harm, because it's very well known that the human genotype affects the way how we metabolize food, all the gene-nutrient interactions. But of course it's quite evident that also this microbiome, the other genome, will play a role here. And we've done a study where we actually showed that indeed the microbiome profiles can be used to predict the metabolic responses in the host, because we showed that basically those individuals who manage to get a clinical decrease of cholesterol levels in the serum during the diet actually had some specific features in their initial microbiota and also in regards how the microbiota responded to these same diets. All in all, there is still a long way to go, because I mean the nutrition science as such is complicated, the microbiome is complicated, and it's a challenge to find the associations between the diet, the microbiome, and the clinical readouts. But it's evident that also there will be a kind of a next generation of probiotics coming from the commensals, so that either the anti-inflammatory or anti-carcinogenic or bacteria that promote the mucosal homeostasis. And I will finish up just by saying that we cannot ignore the microbial implication in our digestion and in the links between nutrition and health. This is an emerging area which of course also complicates the food safety assessment, but as I have shown you, it can reveal novel mechanisms how nutrition affects our health. And in the long term, it will, I think, also contribute to the tailored, personalized nutrition, because as I showed in the last example, basically determining the setup of individuals' microbes before dietary intervention may help to make more efficient and safe products. But more, especially well-designed, sufficiently powered human studies will be needed to progress this field. Thank you.
Thank you very much, Dr. Salonen. We'll now open this up for a few questions and technical clarification. If you have a question, please will you raise your hand in the air so that we can find you? And eventually, can you use the roving microphones, please? When you answer, could you introduce yourself and your affiliation, please? Yeah, go on.
Thank you. Thank you, Tony. Two technical questions,
okay,
if I may.
Alex Liu from Harvard University, Cambridge, Massachusetts. So there is a increasing promotion of paramedicinal product that function as cleansing of the gut. So what is your opinion about this in related to microbiota?
Excuse me, what was the compound called?
The cleansing product, like the beverage that will wash out most of the microorganisms from your gut.
Unfortunately, I'm not familiar with this, so I cannot address the question. I apologize for this.
Okay, thank you. Perhaps you could talk to each other over the coffee time. That would help. Jean.
Two short technical questions. The first one is related to the material which is studied, which is stools, which is a very end product, if I may say so, and which may not reflect the composition of the microbiome at higher stages of the gut. And the other point is that it may not reflect as well the composition of the bacteria at the mucosal level. That's my first question. The second question is, what do you think of the relevance of animal models, mainly rodents, according to the fact that they have a very different, when they are born with an immature gut, whereas humans are born with a mature gut? Thank you.
Yes, very relevant questions. So regarding the usefulness of stool as a representative of the in situ microbiota in the gut, the luminal microbiota, which is excreted as a stool, I think it's relevant in that case when you look, for example, when the interest is on metabolites produced by the gut microbiota, for example, the short-chain fatty acids, because this is the critical mass which produces all these metabolites. But if your target of interest in the study is, for example, the crosstalk, direct crosstalk between the bacteria and, for example, host receptors, then of course a mucosal sample is required, and especially on the children, but in general also in healthy individuals, unfortunately, we basically cannot take biopsies. There are devices being designed that would sample the microbiota that you swallow them, these kind of smart pills, and they are actually quite promising. Regarding the second question, could you very briefly repeat it?
You've done both, I think.
Okay.
Yes, yes. Thank you very much. Any other questions from the audience, please? I have one at the front here. Please.
Thank you. Very interesting presentation. I saw in one of your slides that you have detected cyanobacteria in the gut. These are freshwater bacteria, environmental species. What do you think is the role in the gut microbiome?
I think cyanobacteria are one good example of the bacteria that are frequently detected in the stool, even though it's more likely that they originate, that they are not really inhabitants, but they are transiently there. And this has still not been solved, because basically we cannot differentiate those that, but they're looking for a spot fecal sample, that which bacteria are the permanent colonizers and which, for example, reflect a recent intake of probiotic bacteria. And for that reason, we try as accurately as possible to record what the person has been eating.
Thank you. One last question from one of your panel. Plenary.
And very nice. I have one question. Of course, the composition of the food is extremely important. Another thing that is important as well is the pattern of nutrition. So in other words, when you have natural products like antibacterial peptides, which we produce in our own organism, they are suppressed usually by ingesting food, and they are activated every 4 or 5 hours or so. So then, did you try to correlate, or did anybody try to correlate the intervals of food assumption with the type of food and the possibility of chronic inflammation? So it's pretty obvious that the fanzines, which are produced during a period in which we do not eat, may regulate the microbiota in the intestine, and at the same time, if you do not regulate it well, if you eat continuously, then you may have a state of chronic inflammation, which has been related very much to neurodegenerative diseases or diabetes or other conditions. So the question is that, is there any study that not only compares the composition of the food, but also whether the food is administered at different intervals of time?
Unfortunately, there are no such studies at the moment, but as a separate finding, circadian rhythms are known to affect gut microbiota, so that actually might be one mechanism explaining this. But this is a really relevant point for future research. Thank you.
Okay, well, thank you very much. Thank you, Dr. Salonen. Another round of applause, please.